Good. Everyone all seated? Okay. So, good afternoon, everyone. My name is Shawn Palmer. Eric, I introduce people. I'm a Saxon Scholar here at OMRF, and it's been my pleasure to be part of Dr. Courtney Griffin's lab. Unfortunately, she's not here. I'm going to do my best to make her proud. So, what I'm here to do today is introduce me and Eric's research topic. Basically, we researched regressing blood vessels inside the choroid of patients with age-related macular degeneration. The first question is, what is age-related macular degeneration? Age-related macular degeneration, AMD for short, is an eye disease which affects over 200 million people worldwide and costs annually in the U.S. around $30 billion each year to treat. It is the degeneration of the macula, and for these reasons, me and Eric decided to focus our research over these last 3 weeks on AMD. So, over my time here, we focused on 2 different types of samples: human cross-section samples from the eye bank, specifically AMD donors and non-AMD donors, along with eye samples from a mouse model that Eric developed before I got here. So, the first part, I mentioned earlier that we're studying the choroid. The reason why we're interested in the choroid is because in patients with age-related macular degeneration, there is a distinct regression of vessels inside the choroid prior to damage to the macula. Because of this, we decided that we're going to study the choroid. The choroid itself, as you can see here, is made out of 3 different layers. It's composed of, first, closest to the retina, a layer called the choroid capillaris. These are all these smallest vessels in the choroid. The second layer in the middle is called Saddler's layer. It's composed of medium-sized capillaries. And the final layer, at the very back of the eye, is called Hallis layer. So, me and Eric's research focused on 2 distinct factors. First, something called ETS transcription factors. These factors are present in endothelial cells, and they are necessary for just survival in general. Along with this, we focused on slow flow as a factor. The reason why we focused on these 2 factors in particular was based on another paper released by a member of our lab named Chris Schaefer. He demonstrated that upon administering a ETS inhibitor called YK4279, that these red areas were able to go down. Those red areas represent areas of pathological vascularization, basically unwanted blood vessels. After administering YK4279, he demonstrated that there was a distinct decrease in this area. Along with this, he also demonstrated that the ETS factor, ERG, was also downregulated in constricted blood vessels that he imaged, as you can see at the bottom right here. This is why we focus on the ETS transcription factors and blood flow as our 2 factors. From this, we developed a hypothesis that in order for blood vessel regression to occur, you need 2 things. First, ETS transcription factor downregulation, specifically ERG and FLE1, and slow flow. To start off with, we did 4 different types of staining on our human section samples. The first type of staining is called CD31 staining. It's visualized as these gray areas in the bottom images. These right here are blood vessels. The second type of staining that we did to all samples was something called DAPI staining. This staining stains the nuclear membrane of cells in the choroid, which is visualized as these blue dots in the bottom images. Our third and fourth types of staining were ERG and FLE1 staining. These are represented as the red dots in this image and the next image. This particular image is ERG staining. We can't do ERG and FLE1 staining, we have to do it separately. So, in our samples, we showed that in our patients with AMD, that there was a distinct downregulation of ERG in this example compared to our control samples, along with a lower vascularized area compared to control samples. Furthermore, we see the exact same trend in our FLE1 staining as well, with AMD having a downregulation compared to control and a lower vascularized area. From this point, we moved on to our mouse model. Oh, sorry. Before I get there, after this, we took 3 sets of images from each sample. We did a blind quantification by assigning a random serial number to each sample, and then measuring the average FLE1 expression for our samples. We didn't have enough images of ERG, so we couldn't do that quantification. After we measured the average FLE1 expression, we graphed it, and we show that there is a downregulation of FLE1 inside of patients with AMD. So, after this point, we moved on to imaging our mouse models. One of the biggest issues though is that we use a pigmented mouse model, which means that this little layer called the RPE, the retinal pigment epithelium, is very dark. In figure C, you can see that this is just really dark. It's hard to image this, but it's hard to image this without doing some sort of bleaching. So in order to get better images, we developed a hydrogen peroxide bleaching procedure. To start off, we did 1% hydrogen peroxide for 3 hours with some admittedly harsh pipetting. As you can tell, we kind of destroyed most of our sample for our first attempt. It happens though. We learn from our mistakes. In our second attempt, we upped our hydrogen peroxide percentage, and we did a lot more gentle pipetting. In addition to that, we only did it for 1.5 hours because of our higher percentage. As you can see, we got much better pictures, but we're still not getting a clear amount of choroid. We only get little parts of it, but we still see clear vasculature. This was a step in the correct direction. From this point, we realized that the hydrogen peroxide that we were using was actually going to change the morphology of the eye cup itself. It was making certain parts stick up, which makes it very difficult to mount on a glass slide. So afterward, we did a lot more thorough cleaning of the tissue before we mounted the tissue. After this, we got this beautiful image right here. We got several of these, they look amazing. But based on these images, we noticed 3 distinct vessel morphologies within the mouse choroid itself. First, we have maze-like vessels at the periphery of the choroid. Along with this, we have honeycomb-like vessels also along the periphery. And last but not least, we have finger-like projections from the middle of the choroid. Those are the 3 different areas that we compared our mouse samples to each other with. From this point, we did imaging of eye cups from our mouse model. Our mouse model itself contained a ERG FLE1 flox negative control. What this just means is that it will express ERG and FLE1 regularly. It should have regular vascular development. For our experimental parameter, we used an ERG FLE1 knockout mouse. Basically, what this means is that we can administer a drug called tamoxifen at any point to cause the deletion of the ERG FLE1 gene from endothelial cells. This allows us to downregulate artificially rather than having it be part of some other biological process. In our mouse model, we started administering tamoxifen at 90 days after birth, up there, and for about 5 days using an eyedropper. 5 days afterward, we took the eye cups from the mouse and we imaged them using CD31 staining. Shown here is both the negative control ERG FLE1 flox and our experimental knockout mouse. As you can tell, in both maze-like, honeycomb-like, and finger-like projections, there was not really much difference in our vascularization. We don't know exactly why, but we think there might be 2 possible reasons. First, the eye is a very resource-intensive organ. It requires a lot of blood in order to actually function. A lot of that blood flow is provided for by the choroid, this tissue right here. So because of that, the mouse choroid has one of the highest blood flow per gram of tissue inside the mouse's body, which means that, according to our hypothesis, we needed 2 factors, both ETS factor and inhibition, which we did achieve, along with slow flow. We believe that we did not constrict blood flow enough to the mouse eyes to properly achieve aggression. Additionally, it is possible that we harvested our mouse's eye cups way too early, because we only did it 5 days after administering the tamoxifen. So if we wait a little bit longer, perhaps we can get some better results, some more actual aggression. Overall, during my time at Saxe Scotland, we did two things. First, we were able to demonstrate that in patients with AMD, there was a downregulation of both ERG and FLE1, along with vessel aggression. Additionally, although we were not able to demonstrate that the knockout of the ETS transcription factors can cause regression, we were able to overcome a significant technological hurdle of just bleaching the mouse choroid so we can actually image them. Now, before I go any further, does anyone have questions?
Can you explain why you chose to look at vascularization for this disease?
What is this? We chose to look at vascularization because prior to the macular degeneration in AMD, there was a regression of vessels in the choroid, and that is why we focused on that. Okay. Now, before I step off the stage, I'd like to give a big thanks to Courtney Griffin for allowing me to be part of her lab. Again, she's not here, but her lab is amazing. I love all my fellow lab members. They're all the nicest people I've ever met in my life. And also, I'd like to thank Eric for being a great mentor. He was always present every single day, and he gave me lots of opportunities to grow. Plus, he's very funny. Additionally, I'd like to thank me and Aaron's sponsor, John Saxon, for allowing us to be here. This internship is a great opportunity which would not have been possible without his contribution. And finally, last but not least, I'd like to thank my dad for driving me to and from work every single day.